Hello students, in this video I will discuss about the 12th chapter of second PUC, that is biotechnology and its applications. So in the previous video we have studied about biotechnological principles as well as processes. So this is a continuation of the previous chapter. So let me explain the different applications of biotechnology in various fields. So based on the application, we can classify the biotechnology into different types. The biotechnology applications include therapeutics. We can call it as medicinal biotechnology. Diagnostics, again, it is a part in medicinal biotechnology. And the biotechnology is also applied for the production of genetically modified crops, and it is known as agricultural biotechnology. And in food industry, the biotechnology is applied, so that is for the production of processed food. And even in environmental biotechnology, bioremediation, the waste treatment, as well as the biotechnology is also used for the energy production. So these are the different fields where the biotechnology is applied. So in this chapter we are focusing on the major applications of biotechnology in various fields, particularly application of biotechnology in the field of agriculture, followed by application of biotechnology in the field of medicine, and finally application of biotechnology in animal husbandry. So some of the important examples are described here. Now let us see three important critical research areas of biotechnology. So the biotechnology has a lot of scope in various fields. The three important research areas of biotechnology includes the first one, that is providing best catalyst in the form of improved organism, usually a microbe or pure enzyme. So this is the first area of research in biotechnology. So it says that there is a need to produce one particular enzyme. In biology or in biochemistry, enzyme is nothing but biocatalyst, which activates or which influences a specific type of biochemical reaction. The final intention of genetic engineering or biotechnology is obtaining a particular product, and most commonly that is a proteinaceous product that can be used as a hormone, or that may be the any other kind of drug. So there are different types of proteinaceous products are obtained by the biotechnological processes. So as well as in various kinds of industries, it may be the textile industry or the paint industry, or it may be the food industry, the various kinds of microorganisms or their products such as enzymes are used for various biochemical processes or the biochemical reactions. So we need to provide such a best catalyst, that may be in the form of improved organism, usually a microorganism, or that may be the pure enzyme. So this is the first critical research area of biotechnology. The second one, if the enzyme is produced, that will perform its function only under optimal condition. So providing optimal condition is also the important requirement for the success of the biotechnological processes. So creating optimal conditions through engineering for a catalyst to act is the second research area, and the last one. So we have produced enzyme and we have created such an optimum condition for the proper functioning of enzyme, but the last bit is very important. By using that enzyme or that microorganism, we may produce one particular product under suitable condition, under well furnished laboratory, but the product should be processed properly, that product should be purified properly, that should be added with some preservatives to increase its shelf life, or it should be neatly packed, it should be perfectly packed, and that should be marketed at the end. So all these events are coming under the downstream processing. So improving the downstream technologies to purify the protein or organic compound is also a very important research area, the critical research area of biotechnology. You can expect this for 3 marks. Now, let us say the application of biotechnology in the field of agriculture. We can call it as plant biotechnology, or we can also call it as agricultural biotechnology. Just remember, the plant biotechnology or agricultural biotechnology, that is mainly focusing on improving the qualities of the plants. Qualities in the sense, the properties of the plants, the characteristics of the plants or the plant products. So this is the intention of plant biotechnology or application of biotechnology in the field of agriculture. But before knowing about the application of biotechnology in the field of agriculture, let us see three important options that can be thought for increasing the food production. Nothing but the three important types of agricultural practices. The first one is called as agrochemical based agriculture. Agrochemical based agriculture. It is nothing but by using the various types of chemical fertilizers or chemical pesticides, chemical insecticides, we are culturing the plants. So purely chemical based agriculture. Definitely the yield will be very high. Yield will be very high during the initial period, but slowly there will be decrease in the soil fertility. The excessive usage of chemicals causes air pollution, water pollution, soil pollution. So such kind of the negative aspects are associated with the agrochemical based agriculture. The second type of agriculture, that is called organic agriculture. Organic agriculture is nothing but by using the organic fertilizers, that means biofertilizers or biopesticides, biocontrol agents are used in this type of agricultural practice. So it is purely based on the natural phenomenon, the natural resource, without using any kind of artificially produced or synthetic chemicals. So definitely no harm to the nature, but normally yield is not up to the mark. The yield will be average, not very high. So this is the negative aspect. Lesser yield is the negative aspect, but no pollution, no harm to the nature, that is a positive aspect. So in case of agrochemical based agriculture, yield is high, but at the same time that causes harm to the nature. So that is both the positive as well as negative aspects. So to avoid such kind of problems, to overcome such kind of problems, a new technology, a new method of agriculture is also developed. So that is genetically engineered crop-based agriculture. So this is an advanced method of agriculture, genetically engineered crop-based agriculture. Already we studied about the genetic engineering in the previous chapter. Engineering is nothing but manipulation. Genetic engineering is nothing but manipulation of gene. If you manipulate the gene, the character controlled by that particular gene undergoes change. So that leads to change in the phenotype of an individual. So change may be positive one or negative one. We are selecting the changes with the benefits. So such kind of plants are used in agriculture. So such kind of genetically modified plants, genetically engineered crop plants are used for agriculture. So this is the advanced or the modern type of agricultural practice. So these are the three important types or the three important options that can be thought for increasing the food production. Just observe here, in case of genetically engineered crop-based agriculture, yield is high. At the same time, no much harm, especially no pollution to the in the nature, or there is no pollution. So this is a positive aspect. So we can get many benefits from by using the genetically engineered crop-based agriculture. So now let me explain the important term used in biotechnology, that is GMO. GMO means genetically modified organism. Genetically modified organism. That organism may be the plant, or it may be the bacteria, or it may be the fungi, or even animals. So plants, bacteria, fungi, and animals whose genes have been altered by manipulation are called genetically modified organisms. Genetically modified organism, or GMO. If it is a plant, genetically modified plant. So here we are mainly focusing on the genetically modified plants. And now let us see the applications or benefits or the uses of genetically modified plants. What are the benefits we get from the genetically modified plants in the field of agriculture? See, the genetic modification has made crops more tolerant to abiotic stresses. The plants, they will face many problems. It may be the problem created by some biotic factors such as predators or parasites, or it may be the problems caused because of abiotic factors. The abiotic factors includes temperature, or it may be the pH value, or it may be the salinity. So the various kinds of abiotic factors also influences the growth of a plant. So here, normally the genetically modified plants are produced in such a way that they are more tolerant to abiotic stresses such as cold resistant, drought resistant, salt resistant, heat resistant, temperature resistant plants are produced. So this is a one application, one benefit. Another one, genetic modification has reduced the reliance on chemical pesticides, the usage of chemical pesticides. For example, pest resistant crops helps to reduce the usage of or excessive utilization of pesticides. In the same way, insect-resistant varieties helps to avoid the usage of chemical insecticides. So, by producing the pest-resistant variety, insect-resistant variety, we can reduce the usage of chemicals. So, that is another important application. Third one, genetic modification has helped reduce post-harvest loss. So, even after harvesting a large amount of food will be lost. By developing the genetically modified plants, we can reduce the post-harvest losses. And the another important application, genetic modification has increased the efficiency of mineral usage by plants. So what is this? Say mineral usage. You know, the soil is a source of various kinds of minerals, mineral nutrients. If the soil consists all type of mineral nutrients, the plant should grow normally, and that should show the luxuriant growth. But sometimes, even though there is a presence of all types of mineral nutrients and adequate of amount of water in the soil, the plant may not grow properly. The reason is the plant is not able to absorb the mineral nutrients efficiently. In such case, we need to improve the efficiency of mineral usage by the plants, and that can be achieved by genetic modification. So this prevents early exhaustion of fertility of the soil. And the last one, the last application, genetic modification has enhanced nutritional value of food. For example, golden rice. The golden rice is rich in provitamin A, the precursor of vitamin A or vitamin A. See here, the quantity of food is not important. Quality of the food is important. So that's why we need to produce a food with good nutritive value, and that is possible by genetic engineering. Normally, the genetically modified plant produces nutritive rich food products. So these are the important applications of genetically modified plants. Now let us move to the one of the very important topic from this chapter, that is BT cotton. So this is one of the best example for the application of biotechnology in the field of agriculture. BT cotton is nothing but a genetically modified plant which is resistant to cotton bollworms. So cotton bollworm is an insect. So BT cotton is a genetically modified insect resistant variety. Here the term BT, it refers to a bacterium, that is Bacillus thuringiensis. See, this Bacillus thuringiensis, a bacterium, it produces a special kind of protein called as crystal protein or cry protein. This crystal protein or cry protein is coded by a gene called as cry gene, C-R-Y, cry gene. And these protein crystals are toxic insecticidal proteins. These are the toxic insecticidal proteins. But we should remember, There is no close contact between the insect and the bacteria. But the protein produced by this Bacillus thuringiensis bacteria is found toxic to the insects. So this is the principle behind the production of Bt cotton. The Bacillus thuringiensis consists a gene that is called as cry gene. That cry gene or Bt toxin genes are isolated from them. The specific Bt toxin genes were isolated from Bacillus thuringiensis, and such genes were incorporated into the cotton plant in the embryonic stage. So such cotton plant, the cotton plant developed from such embryo is known as Bt cotton. So Bt cotton consists the gene isolated from the bacterium called as Bacillus thuringiensis. Now what happens in the cotton plant, in the cells of cotton plant, that is the Bt cotton plant, the crystal proteins are produced. The crystal proteins will be formed or produced. So these Bt cotton plants are resistant to cotton bollworms. How they become resistant to cotton bollworms? See, the expression of Bt toxin gene in the cotton plant, in the Bt cotton plant, produces crystal protein in all the body cells, in all the cells, particularly in the leaves. Normally, when the insect, that is the cotton bollworm, is feed on this cotton plant, Bt cotton plant, the plant material will enter into the digestive system of the insect. Just remember, Bt toxin or the crystal protein produced by the host is inactive to the host. I repeat, the crystal protein produced by the bacteria is harmless to the bacteria. If the same protein is produced in the cotton plant, that protein is non-toxic or harmless to the cotton plant. But when the plant material enters into the digestive tract of the insect, the non-toxic or protoxin Bt toxin, that is a crystal protein, undergoes chemical change that leads to the formation of a toxic Bt toxin. The reason is under alkaline condition. There is an alkaline condition in the digestive tract or gut of the insect. Under that alkaline pH, the inactive toxin is converted into active form of toxin. Now, that will solubilize an alkaline pH of the gut, which solubilize the crystals, and that causes the formation of small openings or pores in the epithelial cells of the lining of the digestive tract. You know, digestive tract, the lining of the digestive tract is made up of epithelial cells. The toxic chemical produced by the conversion in the alkaline pH condition causes the formation of openings in the epithelial cells, and it results in the intake of water by the epithelial cells. The cell swelling takes place and such cells will burst. The bursting of epithelial cells in the digestive tract lining causes the death of the insect. So the activated toxin binds to the surface of mid-gut epithelial cells and create pores, and that leads to the cell swelling and lysis. Lysis means breakdown, and finally leads to the death of the insect. So this is the production of Bt cotton, the principle behind the production of Bt cotton. The type of cry genes. There are different types of cry genes. Based on the insect harming the plant, we can use the different types of cry genes. The choice of the genes depends on the crop and the targeted pest, as most Bt toxins are insect group specific. Bt toxins are insect group specific. There is a particular Bt toxin harmful to a particular group of insect. So that's why they are very specific in their toxicity. The toxin is coded by a gene called as Cry1AC. The toxin is coded by a gene that is Cry1AC named Cry. So that is the name of the gene. There are a number of them. For example, proteins encoded by genes Cry1AC and Cry2AB control the cotton bollworms. That of Cry1AB controls corn borer. So cotton bollworms are controlled by Cry1AC and Cry2AB genes because the proteins encoded by them are harmful to bollworms. And the protein produced by Cry1AB is harmful to corn borer. So this is about the types of Cry genes. So just remember, the Bt toxin is produced by bacterium called Bacillus thuringiensis. Here Bt stands for Bacillus thuringiensis. Bt toxin gene has been cloned from the bacteria. It is cloned from the bacteria, isolated from the bacteria, and being expressed in plants, introduced into the plants, and such gene undergoes expression in the plants to provide resistance to insects without the need for insecticides. So we can reduce the use of insecticides. In effect, created a biopesticide. So the next one, some of the strains of Bacillus thuringiensis produce proteins that kill certain insects such as lepidopterans. For example, tobacco birdworm as well as armyworm are the example for lepidopterans, as well as coleopterans, example beetles, and dipterans, example flies and mosquitoes. So this is regarding the production of Bt cotton. At the same time, the various other genetically modified plants are also developed. In addition to Bt cotton, Bt corn, Bt rice, Bt tomato, Bt potato, as well as Bt soya bean are also produced by genetic engineering. Now, let me explain another very important type of genetically modified plant. They are called pest-resistant varieties. The production of pest-resistant varieties by a specific method that is called as RNAi method. So RNAi method means RNA interference method. The simple explanation for this method is given in the textbook. I will explain as it is given in the textbook. Okay, so it has many other additional steps. All the steps are not mentioned. I will explain as it is given in the textbook. So you should remember one particular pest called as Meloidigain incognita. It is a nematode found in the soil. So this causes harm to the roots of certain plants, especially to the plants such as tobacco. It causes infection to the roots, and that results in decrease in the yield, reduced yield. So to kill such kind of nematode pests, we can use the pesticides. But in another way that causes harm to the nature, it causes soil pollution or water pollution. To avoid the usage of such kind of chemicals, we have developed genetically modified plant which is resistant to this particular pest, that is a nematode called Meloidigain incognita. So the methodology or the technique for the production of pest resistant variety is called as RNAi method or RNA interference method. See, RNAi actually takes place in all eukaryotic organisms as a method of cellular defense. So this is a natural phenomenon, takes place in all the eukaryotic organisms. So here, this principle is used for the development of pest-resistant varieties. See, this method involves silencing of a specific mRNA due to a complementary double-stranded RNA molecule that binds to and prevents translation of the mRNA, and that process is called as silencing. So I repeat, in this method, a special type of RNA molecule is produced, that is a double-stranded RNA molecule, by using a specific gene, and that double-stranded RNA is used for the silencing of another RNA. That another RNA is nothing but pathogenic RNA, pathogenic mRNA. So this is the principle behind this technology. The source of this complementary RNA, the complementary RNA means the double-stranded RNA. The source of this double-stranded RNA, it may be the cell which is infected by a viruses having RNA genomes. RNA genome, ribovira. The virus with RNA genome is called as ribovira. So ribovira infected cells can be used as a source of this complementary RNA or the double-stranded RNA. Or we can use the mobile genetic elements that is called as transposomes or the jumping genes as a source of this particular double-stranded RNA. And normally such kind of viruses, they will, RNA viruses, they will replicate via RNA intermediate. So here, the double-stranded RNA is coded by a particular type of gene that is particularly named as nematode-specific gene. That is called as nematode-specific gene. Such nematode-specific genes which are coding for a double-stranded RNA are introduced into the host plant using agrobacterium as vectors. So the nematode-specific genes are nothing but the special kind of genes coding for a double-stranded RNA are introduced into the host plant by the help of a vector called as agrobacterium. The introduction of DNA, that is the nematode-specific gene, the introduction of nematode-specific gene or DNA was such that it produced both sense as well as antisense RNA in the host cells. Normally, in the normal cases, DNA, that is a double-stranded DNA, produces single-stranded RNA by the process called as transcription. But here it is a special case. Here, the nematode-specific gene produces double-stranded RNA. So one strand is a sense strand, another one is an antisense strand. Normally, these terms are used in case of DNA. In the transcriptional unit, one strand is the sense strand, another one is antisense strand. So always the antisense strand directs the synthesis of RNA. Here, both the strands are involved so that the complementary RNA are produced. If the two RNA strands are complementary to each other, they will undergo hybridization. See, these two RNA strands being complementary to each other formed a double-stranded RNA, dsRNA, that initiated RNAi and thus silenced the specific mRNA of the nematode. So the special kind of RNA, that is a double-stranded RNA produced by this mechanism in the host plant body, that means in the genetically modified plant body, silences the expression of mRNA of the pathogen, mRNA of the nematode. See, for this we should know one important point. Always, the pathogen will cause the disease or pathogen will show the disease symptoms in the host body only if its genetic material is expressed in the host cell. But you should remember there are two steps in the genetic gene expression. One is called transcription followed by translation. During transcription, RNA will be formed. And that RNA, particularly mRNA, that undergoes translation for the production of pathogenic proteinaceous product. If that protein, pathogenic protein is produced, then only there will be the development of disease symptoms. So here what happens, the mRNA is produced by the pathogenic genetic material. But that mRNA of the pathogen will be silenced. That will not undergo translation so that there is no production of pathogenic protein, and that's why there is no development of disease. So the parasite could not survive in transgenic host. The transgenic plant therefore got itself protected from the parasite. So this is about the production of RNAi methodology for the production of pest resistant varieties. So here the simple explanation is given in the textbook. This is not so simple. Some additional points are also there, but such points are not given in the textbook. That's fine. I will not discuss here. Okay, by this I will stop this chapter here. In the next video, I will continue the remaining topic.